We are going to have a brief discussion about two topics. Number one, gastric secretion. Number two, pancreatic secretions. Number two, pancreatic secretions. Alright, now let us take up the very first topic, doctors. Let's take up the very first topic. Let's take up gastric secretions. At the outset, be aware that when it comes to gastric secretions, we are going to have two kinds of gastric secretions. The first one is going to be secretion of hydrochloric acid, and the second one is going to be secretion of gastric enzymes. Hydrochloric acid as well as gastric enzymes. Now, to begin with, you need to be familiar about the glands which are present in stomach and the way with which stomach secretes its content. At the outset, when it comes to secretory apparatus of stomach, doctors, the first and foremost structure you need to be familiar with is surface mucosal epithelial cells. Surface mucus producing epithelial cells, doctors. Now, when it comes to mucus epithelial cells, we are going to see as mucus producing epithelial cells all throughout the stomach surface, starting from cardia all the way to body of stomach, pylorus. You do have mucus producing epithelial cells lining the entire surface of stomach. Alright, so at the first outset, be familiar with that mucus producing epithelial cells are seen all throughout the gastric mucosa. And this mucus producing epithelial cells keeps on producing mucus, and this mucus production is going to be at a level of about 1 millimeter thick. The mucus production is going to be at a level of 1 millimeter thick. Alright, that's the first and foremost mucus producing structure you need to understand, doctors. Yeah, that's mucus producing structure. After which you need to be familiar about gastric glands. Now, when it comes to gastric glands, we have two kinds of glands, doctors. The first one is going to be oxyntic glands, which are also called as gastric glands. Oxyntic glands, which are also called as gastric glands. Number two, there is going to be pyloric glands. There are two kinds of glands in stomach. The one is oxyntic glands, also called as gastric glands, and the second one is pyloric glands. What are the difference? Let us take up oxyntic glands first. Oxyntic glands are seen about 80% of gastric mucosa. 80% of gastric mucosa do have oxyntic glands. It extends from cardia of stomach, it goes all the way to body of stomach, and it is also seen very close to antral regions. Oxyntic glands are seen from cardia, body, as well as very close to antral regions. So, as I just told you, 80% of gastric mucosal surface do have oxyntic glands. Few key important cells of oxyntic glands are, there is going to be surface mucosal epithelial cells, mucus producing epithelial cells, and this cell is going to produce mucus. Along with that, there is going to be parietal cells. Parietal cells are also called as oxyntic cells. Because of the presence of oxyntic cells, these glands are called as oxyntic glands. Parietal cells or oxyntic cells produces, number 1, hydrochloric acid. Number 2, they also produces intrinsic factor. They also produces intrinsic factor. So parietal cells or oxyntic cells produces hydrochloric acid as well as intrinsic factor of Castle. Now, we do see other cells, that is enterochromaffin-like cells. Enterochromaffin-like cells produces histamine. Enterochromaffin-like cells produces histamine. The cells which belong to oxyntic glands are predominantly involved in secreting hydrochloric acid, intrinsic factor, as well as histamine, doctors. These three secretions are the most important secretions coming out of oxyntic glands. And we are going to see a very important cell called the chief cells. Chief cells do produce an enzyme called as pepsinogen. So these are the secretions coming out of oxyntic glands, also called as gastric glands. Remember, first important is hydrochloric acid, intrinsic factor of Castle, histamine, as well as pepsinogen. These are the secretions coming out of oxyntic glands. Coming to pyloric glands. Pyloric glands are present in just 20% of gastric mucosa. Pyloric glands are present only in 20% of gastric mucosa. That too, they are very exclusively located close to pylorus. These pyloric glands are not present in cardia, not present in fundus, body or antral region. They are located very close to pyloric region. The importance is pyloric glands do have mucus producing cells, more abundant mucus producing cells. More abundant mucus producing cells that keep secreting mucus. And apart from mucus producing cell, pyloric glands do also have G cells. And G cells are involved in production of gastrin. G cells are involved in production of gastrin, doctors. So pyloric glands are involved in production of mucus as well as gastrin. Now if you see gastrin which gets secreted from pyloric glands situated in pylorus, this gastrin enters bloodstream and acts as a hormone to trigger parietal cells in order to enhance gastric acid production. On the contrary, pyloric glands produces enormous amount of copious mucus. The reason why these glands produces mucus is the gastric acid that got produced in oxyntic glands situated in cardia, body and antrum, the acid must be neutralized. Only then the content that gets into the duodenum crossing the pyloric sphincter will go with optimal pH. You know very well, duodenum is an alkaline environment region. Pancreatic enzymes as well as bile that get secreted into duodenum will act only in alkaline pH, for which duodenal lumen must be kept alkaline. Under no circumstances duodenal lumen can become acidic. Hence, pyloric glands which are located at the pylorus keeps producing enormous amount of mucus. By the time the gastric acid reaches pylorus, this mucus coats the gastric acid and ensures there is not going to be any acidity to the kind that crosses pylorus to enter into duodenum. That's the ultimate reason why pyloric glands are abundantly producing mucus. So these two, alright doctors, these two glands are first and foremost important points you got to understand when it comes to gastric secretions, oxyntic glands as well as pyloric glands. Now with this basics, let us move on to discuss about the mechanism with which gastric acid is getting secreted. Let's take up the mechanism of gastric acid secretion. Let us take up parietal cells here. And this parietal cell is going to keep producing gastric acid, for which these cells are dependent on blood supply. For effective production of gastric acid, these cells are dependent on blood supply. These vessels are the one that's going to continuously supply chloride for the production of hydrochloric acid. So let us start with movement of chloride from the blood vessel into the interstitium. Once chloride is taken into the interstitium, it is now ready to get into the parietal cell to be secreted into the gastric lumen. Let's consider we have gastric lumen on one end and interstitium on the other end. Inside the parietal cell, we are going to observe water molecules, and this water molecule is going to split up into hydroxyl ion as well as hydrogen ion. The hydroxyl ion thus formed is going to combine with carbon dioxide to form bicarbonates. It is going to combine with carbon dioxide to form bicarbonate. And the bicarbonate thus got formed is going to be exchanged. There is going to be movement of bicarbonate out of the cell for exchange of chloride into the cell. This is exactly how the chloride that got out of the blood vessel is taken into the parietal cells. So we are seeing movement of chloride into the parietal cells, doctors. The chloride thus got into the parietal cell is now taken into the gastric lumen. The chloride that is entering the parietal cell is going to be secreted into gastric lumen. That's how we are going to get chloride ion secretion separately. The hydrogen that formed by breakdown of water is going to be exchanged in an active transport manner by primary active transport. We are going to see transport of hydrogen ion for exchange of potassium. And this transporter is called hydrogen potassium ATPase. The transporter is called hydrogen potassium ATPase, doctors. By breakdown of a molecule of ATP, hydrogen is secreted into the lumen and potassium is reabsorbed into the parietal cells. This pump is also called as proton pump. Proton pump inhibitors like pantoprazole, omeprazoles and all are going to act and inhibit the proton pump, thereby they ultimately block hydrogen ion secretion out of the parietal cells. Once hydrogen comes separately, chloride comes separately in the gastric lumen, they both combine. In the gastric lumen, they both combine together and net formation of hydrochloric acid happens in gastric lumen. So there is going to be formation of hydrochloric acid in the gastric lumen only. The cell are not going to produce fully formed hydrochloric acid. If you can see, for every hydrogen ion that gets secreted into the gastric lumen, we are observing one bicarbonate ion that is getting reabsorbed into the blood vessel. Alright, bicarbonate ion is getting taken into the blood vessels. For every hydrogen that gets secreted into the gastric lumen, one bicarbonate is reabsorbed into the blood vessels, doctors. This is exactly how parietal cells keeps producing gastric acid. This gastric acid production is influenced by certain factors. Certain factors are going to trigger parietal cell acid production, and there are going to be some factors which inhibits acid production. Now let us start with the factors that trigger parietal cell acid productions. The most important factor among all is going to be a hormone called Gastrin, which is going to trigger parietal cell acid production, doctors. Let's take up Gastrin. Gastrin is produced by cells in the pyloric glands called G cells. We just observed G cells, isn't it? We have G cells in pyloric glands at the pyloric region which produces gastrin. Gastrin, even though it gets produced, it is not going to exert its action straight away. Gastrin do have two kinds of action on parietal cells. Number one, the predominant way of action is gastrin is going to excite a cell called enterochromaffin-like cells. We have already discussed about these cells, doctors. Hope you could able to remember. All right. So we have enterochromaffin-like cells which produces histamine. There are enterochromaffin-like cells, and these ECL cells is going to produce histamine, and histamine is going to excite parietal cells through a receptor called H2 receptor. Very important, histamine is going to act via H2 receptors, doctors. Acting via H2 receptors. And this H2 receptor is going to excite parietal cells, and this action of histamine on parietal cells is the predominant action with which acid secretion happens. Now, what influences histamine release? Is it enterochromaffin-like cells straight away? No, absolutely not. Enterochromaffin-like cells has to be activated by gastrin coming from G cells. Now, apart from gastrin activating enterochromaffin-like cells, they themselves do have an action on parietal cells, and this gets executed by another receptor called cholecystokinin B receptor. That's again a very important point. Gastrin straight away excites parietal cells through cholecystokinin B receptor. It also triggers enterochromaffin-like cells in order to produce histamine and activating parietal cells by H2 receptors. Out of these two mechanisms, the most predominant mechanism of action of gastrin is going to be activating ECL cells to release histamine. Now, the enterochromaffin-like cells are activated not only by gastrin coming from G cells, but there is also going to be activation of these cells by vagus nerve. The 10th cranial nerve, vagus nerve, is going to activate enterochromaffin-like cells through neurotransmitter acetylcholine. The vagus nerve is going to activate G cells as well. But one slight difference, the neurotransmitter of vagus nerve activating G cells is not acetylcholine. And the neurotransmitter is going to be gastrin releasing peptide. The neurotransmitter is going to be gastrin releasing peptide, doctors. Alright, fine. So that's gastrin releasing peptide, the neurotransmitter of vagus nerve influencing G cells, and acetylcholine influencing enterochromaffin-like cells to produce histamine. Now, apart from these two actions of vagus nerve straight away influences vagus nerve through muscarinic receptor type 3. Through muscarinic receptor type 3, vagus nerve straight away influences parietal cells to produce gastric acid, and again the neurotransmitter in this pathway is going to be acetylcholine. So the stimulation of parietal cells happens by three different mechanisms. Number one, acetylcholine from vagus. Number two, histamine from enterochromaffin-like cells. Number three, gastrin from G cells. Out of these three types of excitation, the predominant excitation is by histamine coming from enterochromaffin-like cells. Now, these are all the excitatory pathways, doctors. These are the excitatory pathways for parietal cells. Parietal cells are getting inhibited by. Let us take up inhibitory factors, people. Now we are going to experience three kinds of molecules. Number one is going to be somatostatin. D cells. These D cells are located in multiple regions. Number one, we are going to see D cells in oxyntic glands itself. D cells are seen in oxyntic glands. D cells are seen in pyloric glands. D cells are seen in duodenum. D cells are seen in pancreas. D cells are seen in oxyntic glands, pyloric glands, duodenum as well as pancreas. These D cells releases somatostatin, and the predominant inhibitory factor for gastric acid production is indeed somatostatin, doctors. Alright, number 2. Apart from somatostatin, we are going to see two extremely important hormones involved in blocking gastric acid production. The first and foremost among which is secretin. Secretin has got a direct inhibitory action on parietal cells acid production. Apart from the action of secretin, there is also going to be inhibition of parietal cells by cholecystokinin. Remember, we already saw cholecystokinin A receptor, cholecystokinin B receptor, which is utilized by gastrin, isn't it? Now, cholecystokinin is going to act on cholecystokinin A receptor. Cholecystokinin A receptor, and this is going to ultimately inhibit gastric acid production, doctors. So that's how gastric acid is getting influenced by stimulating factors as well as inhibitory factors, and that's gastric acid synthesis. Now, apart from hydrochloric acid, you guys know very well, parietal cells are also involved in synthesizing intrinsic factor. And intrinsic factor is going to be useful in reabsorbing vitamin B12. Please remember, even though intrinsic factor is getting produced in stomach parietal cells, vitamin B12 is actually getting reabsorbed in terminal ileum. In order to enhance ileal reabsorption of vitamin B12, stomach keeps producing intrinsic factor from its parietal cells. So, in case of chronic gastritis, atrophic gastritis, when the parietal cell mass is getting suppressed or reduced, apart from the patient developing achlorhydria, the patient also goes for pernicious anemia because of lack of vitamin B12, because of deficiency of intrinsic factor of Castle. Alright, that's parietal cells for you. Now, apart from parietal cells producing intrinsic factor, we also have chief cells. Chief cells are involved in production of a proteolytic enzyme called pepsinogen. Now, at the outset, please understand, pepsinogen is a proteolytic enzymes. And this enzyme, pepsinogen, is designed to break proteins. They are designed to break proteins. Now, pepsinogen gets activated. Pepsinogen is an inactive enzyme. Alright, please remember, it is an inactive enzyme. Any inactive enzyme is also called as zymogens. And the cells that produces inactive enzymes, the cells that produces zymogens, are called as zymogen producing cell. I repeat, this has been asked many times in entrance exams. The enzyme in inactive form is named as zymogens. The enzyme in inactive form is named as zymogens. Pepsinogen is a zymogen because it gets synthesized as inactive molecule. In the presence of hydrochloric acid, pepsinogen gets converted to pepsin, doctors. It gets converted to active molecule called pepsin. And pepsin is going to be involved in protein breakdown. Pepsinogen is converted to pepsin in the presence of hydrochloric acid in the acidic pH, and pepsin is going to digest proteins in the stomach. You know very well, the digestion of carbohydrates begins in oral cavity. Because of presence of an enzyme called tyalin, salivary alpha amylase. Similarly, digestion of proteins begins in stomach because of the enzyme called pepsin, which gets produced as an inactive molecule of form called pepsinogen. In the presence of gastric acid, it gets activated to pepsin, my friends. That's lovely. Now, once pepsin is being formed, there is going to be a narrow window for pepsin to act. Alright, pepsin needs acidic pH to act, doctors. When it comes to the action profile of pepsin, pepsin needs acidic pH to act, and hence pepsin acts only at a pH of 1.8 to 3.5. Pepsin will act to break down protein molecules only at the pH of 1.8 to 3.5. Beyond this range of pH, pepsin does not act and pepsin becomes inactive. So within very few minutes, pepsin can be easily inactivated if the pH is taken out of the range. People. So that's the secretions of stomach when it comes to chief cells, parietal cells, as well as the mucous cells. Now you also have to understand the hormone coming out of pyloric glands. When it comes to pyloric glands, people, we see that pyloric glands do have two kinds of cells. One is mucosal cell that produces mucus. The second one is G cells. G cells produces gastrin. It's a well orchestrated secretory mechanism. If you look at the processes involved in gastrin secretion, you'll be literally amazed, doctor. It's so beautiful. All right. Now, the primary trigger for production of gastrin comes by vagus nerve. All right. So let us take up the very first trigger coming from vagus nerve. And vagus nerve releases gastrin releasing peptide. We just discussed gastrin releasing peptide in acid secretion. You remember, we have a neurotransmitter called the gastrin releasing peptide, which is the neurotransmitter of vagus nerve, isn't it? So this vagus nerve produces gastrin releasing peptide, which is going to act on G cells and ultimately leads to gastrin production. That's trigger number 1. Trigger number 2, peptides. Peptides as well as amino acids. Peptides as well as amino acids in the content of food that reaches stomach is also going to trigger G cells. These two are the triggering factors for G cells, doctors. Remember, one is going to be vagus nerve, the neurotransmitter, gastrin releasing peptide. The second one, peptides and amino acid in the content of bolus, the one that we eat. As a consequence of G cell activation, there is going to be production of the hormone gastrin. Gastrin acts as a hormone involved in multiple functions. The functions of gastrin are: number 1, gastrin is involved in stimulating acid productions. Stimulating HCl production by parietal cells. Number 2, gastrin relaxes. Gastrin relaxes pyloric sphincter. And gastrin promotes gastric emptying. That's second action. Alright. Number 1, it stimulates acid production. Number 2, it relaxes pyloric sphincter and thereby it promotes gastric emptying, doctors. Action number 3. Gastrin increases growth of gastric mucosa. Gastrin increases the growth of gastric mucosa Number 4, to very little extent, gastrin increases the tone and thereby gastrin contracts lower esophageal sphincter. Alright, gastrin increases the tone and contracts lower esophageal sphincter, doctors. Fine, these are all the actions of gastrin. Now, if you consider gastrin producing cells are not only going to be present in stomach, there are gastrin producing G cells which are located in duodenum as well as pancreas. Now, we have observed the stimulating factors for G cells. Let's also get to understand inhibitory factors for G cells. G cells are getting inhibited by number 1, somatostatin. Somatostatin inhibits G cells. Number 2, secretin, the hormone coming from duodenum. The hormone secretin coming from the duodenum inhibits G cells, doctors. So G cells are triggered by two factors, one vagus nerve, the second one amino acids and peptides in the stomach. It is getting inhibited by somatostatin as well as secretin. These are all the gastric functions as well as gastric secretions, doctors. Now, we finish gastric secretion with one very important point, that's phases of gastric acid secretion. Now, we are seeing three phases of gastric acid secretion, doctors. The first one is cephalic phase. The second one is gastric phase. And the third one is intestinal phase. Alright, people, cephalic phase, gastric phase as well as intestinal phase. Cephalic phase of acid secretion is because of sight of the food, smell of the food, taste of the food, as well as thought of the food. Because it is purely cerebral, alright, sight, smell, taste, thought, this occurs, the acid production occurs well before the content reaches stomach. Before even the content reaches stomach, there is going to be production of gastric acid, and this happens at a 10% rate. 10% of acid secretion, 10% of acid secretion is purely cephalic. Before even the food reaches stomach, there is going to be production of gastric acid. The whole cephalic phase of gastric acid secretion is mediated by vagus nerve. The whole cephalic phase of gastric acid secretion is mediated by vagus nerve, doctors. That's very important. Coming to gastric phase, this is because of presence of food in the stomach. Because food is present in the stomach, alright, there is going to be activation of G cells. There is going to be activation of G cells, and G cells are going to in turn trigger production of gastrin, and you know very well, because gastrin is getting released, there is going to be acid secretion. By this way, ultimately through gastrin, if acid secretion are increased, the phase of acid secretion is called as gastric phase of acid secretion. This involves about 60% of acid secretion, doctors. The gastric phase of acid secretion is about 60%. Please remember, cephalic phase is 10%, gastric phase is 60%. Alright, even cephalic phase go all the way up to 10 to 30%. The cephalic phase, maximum of cephalic phase happens up to 30%. Alright, gastric phase is 60%. Coming to intestinal phase. Intestinal phase is very minimal. Alright, because of presence of acidic contents in the intestines. Alright, there are going to be some G cells in duodenum. Some G cells in the duodenum. Alright, so the peptides and amino acids, you know very well, peptides and amino acids do have a direct influencing effect on G cells of duodenum, isn't it? So in the duodenum, if there are peptides and amino acids, they go and excite G cells in duodenum. As a consequence, this G cell releases gastrin, and the gastrin is going to enhance acid production. So increase in gastric acid production because of G cells getting activated in duodenum is called as intestinal phase of acid secretion, and this ranges up to 5 to 10%. Intestinal phase of acid secretion ranges up to 5 to 10%, doctors. So these are all the three phases of acid secretion: cephalic phase, gastric phase, as well as intestinal phase. Alright, that completes our gastric acid secretion. Let us take up the second topic of today's discussion, pancreatic secretion, doctors. Now, in this discussion, we are going to restrict ourselves to the enzyme production from pancreas, not the hormones. We are not going to involve in the endocrine functions of pancreas. We are not going to talk about insulin production or glucagon production. The focus of this video is all about enzymatic productions from pancreas. When it comes to pancreatic secretions, we are going to observe two kinds of secretions from pancreas. Even though they are enzymes, the exocrine part of pancreas produces two kinds of secretions. Let us start with the acinar cells. Alright, we have pancreatic acini that produces enzymes. So let me take this as pancreatic acini. This pancreatic acini is going to be drained. Pancreatic acini is going to be drained by pancreatic duct. This is pancreatic duct, doctors. And this pancreatic duct is going to join with hepatic duct before it enters into the second part of duodenum. You know very well, there is going to be sphincter of OD governing an ampulla called the ampulla of Vater, where the hepatopancreatic duct opens into the second part of duodenum jointly. Now this duct do have its own epithelial cells called ductal epithelial cells. Now we are going to see two kinds of secretions here, people. Now the pancreatic acini is going to release digestive enzymes. Pancreatic acini is going to release digestive enzymes, and digestive enzymes are enzymes belonging to digestion of carbohydrates, fats, as well as proteins. Before even we get to the digestive enzymes, let's understand on a very common scale, the pancreatic acini are going to be excited by a hormone which is going to trigger pancreatic acini to produce digestive enzymes, and this hormone is nothing but cholecystokinin. Cholecystokinin triggers pancreatic acini to produce digestive enzymes. On the other hand, when it comes to ductal cells, let me take up one ductal cell for a detailed understanding, doctors. For a detailed understanding, let me take up a ductal cell here. So let's take up a ductal cell. In the cytoplasm of ductal cells, we are going to observe water molecules which is going to combine with carbon dioxide. The combination of carbon dioxide and water is going to form carbonic acids. And this carbonic acid is going to split into hydrogen as well as bicarbonates. The bicarbonate thus formed in the ductal cells are going to be secreted into the ductal lumen. It is going to be secreted along with sodium ion. So there is going to be secretion of sodium and bicarbonate in lumen of pancreatic duct. In the lumen of pancreatic duct, there is going to be secretion of sodium and bicarbonate, while the hydrogen ion is going to be reabsorbed back into the blood vessels. Alright, on the interstitial sites, there is going to be removal of hydrogen ion, and this hydrogen is going to be exchanged. For sodium. So on the interstitial side, we are going to see sodium hydrogen exchanger. This transporter is called as sodium hydrogen exchanger. Sodium hydrogen exchanger is going to take hydrogen ion out of the cell and sodium ion into the cell. Together there is going to be secretion of sodium as well as bicarbonates into the lumen. So if you clearly see, the pancreatic acini are going to produce digestive enzyme, while the lumen keeps producing bicarbonates. Ductal cells keeps producing bicarbonates into the lumen. So the ductal cellular secretion of bicarbonate into the lumen is excited by another different hormone called as secretin. So you have two kinds of hormones, people. One is cholecystokinin that triggers production of pancreatic digestive enzyme, and secretin that triggers production of bicarbonate from the ductal cells. These are all the two major secretions of pancreas. One is enzymes and the second one is bicarbonates. And if you consider the digestive enzymes coming from pancreas, the first and foremost digestive enzyme are for digestion of carbohydrates. For digestion of carbohydrates, pancreas keeps producing pancreatic alpha amylase. It produces pancreatic alpha amylase, which is going to act on complex polysaccharides and breaks down complex polysaccharides into disaccharides. Pancreatic alpha amylase acts on complex polysaccharides. Complex polysaccharides are also called as starches. At the end of this digestion, these starches are getting converted into disaccharides. They get converted to disaccharides. That's carbohydrates and pancreatic alpha amylase. When it comes to digestion of lipids, for digestion of lipids, there are going to be pancreatic lipase. And there is going to be pancreatic colipase, cholesterol esterases. cholesterol esterase as well as phospholipases. These are all the 4 enzymes secreted by pancreatic exocrine part in order to break down lipids. That's enzymes for lipids, doctors. And the last part, enzymes for protein digestion. Enzymes for protein digestion. Pancreatic enzymes for protein digestions are number 1, trypsin. The very important enzyme is trypsin, chymotrypsin, number 3, carboxy polypeptidases. Trypsin, chymotrypsin and carboxy polypeptidases, these are all the enzymes secreted by pancreas for protein digestion. Again, if you look at these enzymes, all these enzymes are produced as zymogens. There is going to be production of trypsinogen and chymotrypsinogen, as well as carboxy polypeptidases. Trypsinogen, chymotrypsinogen, carboxy polypeptidases. Trypsinogen will get converted to trypsin. It gets activated to trypsin in the presence of an enzyme called enterokinase, which is produced by brush border cells of intestines. Intestinal enterocytes produces an enzyme called enterokinase, which activates trypsinogen into trypsin. Once trypsin is formed, trypsin is a very versatile enzyme. Trypsin do have a excitatory action on itself. Trypsin keeps activating more trypsinogen to get converted to trypsin, doctors. So trypsin is such a versatile enzyme. Apart from activating the inactive trypsinogen to trypsin, it is also involved in activating chymotrypsinogen. To chymotrypsin. And procarboxy polypeptidases into carboxy polypeptidases. Again, this is done by trypsin, doctors. So once trypsin is formed, the subsequent conversion of inactive enzymes into active enzymes are made by the same trypsin molecule itself. Such a versatile enzyme. It is because of this very vigorous versatile activity, there are very high chances that the trypsin might break the pancreatic cells itself. There can be a very high chance of pancreatic autolysis by its own trypsin molecules, which is why the cell that produces trypsin, in order to break down proteins that are present in the diet, the same cell are also going to produce a very important enzyme called a trypsin inhibitor. Alright, so that's very important. Let's take up a pancreatic acini here. Pancreatic acini, and we are going to have a pancreatic duct. Pancreatic acini, as they start producing the enzyme trypsinogen, the same cell which is also going to produce another important enzyme called trypsin inhibitors. And these trypsin inhibitors are so important in order to prevent autoactivation of trypsin. Thereby, it is very important to prevent breakdown of pancreatic cells itself. So prevents autoactivation of trypsin and prevents breakdown of pancreatic cells. These are all done by trypsin inhibitor enzyme produced by the very same cell that produces trypsin, doctors. Now, you know very well trypsin needs an enzyme called enterokinase to get it activated, and enterokinase is produced by brush border enterocytes, isn't it? The cells that lines the intestinal lumen do have brush borders, and there is going to be an enzyme called enterokinase, and this enterokinase is going to convert the inactive trypsinogen into active trypsin. So till this conversion takes place in the lumen of duodenum and jejunum, trypsin inhibitor ensures there is not going to be any activation of trypsin inside the pancreatic duct or inside the pancreatic acini as well. In case of pancreatic ductal problems like pancreatic duct occlusion, pancreatic stones, in these conditions there is going to be enormous accumulation of zymogens in the pancreatic duct. Under these circumstances, there is going to be a overwhelming activity of pancreatic enzymes, and this trypsin inhibitor cannot be able to block the conversion of inactive trypsinogen into active trypsin. In case of such pancreatic duct blocks, trypsin becomes activated. All hell broke loose. The active trypsin is going to vigorously digest pancreatic cells, leading to massive, severe, terrible pancreatitis. To the point that it might even go for worsening. Alright, it might even go for worsening, doctors. This might even lead to, massive pancreatitis leading to shock. There can even be associated with shock, doctors. That's gastric acid secretion as well as pancreatic secretions for your discussion. Fine, people. So yeah, of course you are going to have stem cells. Stem cells are seen in intestinal crypts. Stem cells are seen in intestinal crypts, and these stem cells are nothing but Paneth cells. They are seen in intestinal crypts, doctors. Alright. Now this Paneth cell or stem cell is not seen in gastric pits. Please remember, they are not seen in gastric pits. Rather, they are present in intestinal pits. Fine, that's one doubt. And during cardiac action potential, phase 0, sodium in, phase 1, potassium out, phase 2, potassium out, and calcium in, phase 3, potassium out. So where does this potassium come from, and where does sodium and calcium go? All right, that's a very good question. Now, Dr. Gupta, understand this. Now, when it comes to the ionic transfers, during depolarization, sodium comes into the cardiac cells. From extracellular fluid. Similarly, calcium comes into the cardiac cells from extracellular fluid. These two events happens during depolarization. Now, during repolarization, potassium comes out of the cell to extracellular fluid. Please remember, Potassium comes out of the cell to extracellular fluid, while sodium and calcium, they are pumped back to extracellular fluid. They are pumped back to extracellular fluid, doctors. That's during repolarization. All right, so that's our doubts for today. Now I hope you got these doubts cleared, and you must be familiar with gastric secretions and pancreatic secretions, doctors. That completes the end of our discussions, and we are still live in our DBMCI eGurukul Telegram group. If you need any doubts at any point of time, DBMCI eGurukul will be happy to address all these doubts, doctors, and I'll make sure your physiology doubts are getting cleared. Best wishes, stay positive, do your exams well, keep rocking. Good night, doctors. That completes our end of the discussion. Thank you very much and good night.